Guys, my name is Amrai Parks, and today we will be discussing the role of mitochondrial ATP synthase in cancer. And so, before we get into the bulk of the paper, it's important to understand the function of the mitochondrial, which is basically to produce ATP. We know that the production of ATP is used in many metabolic pathways. And for the sake of this paper, we will be discussing ATP synthase's role in cancer. So that leads me to the topic of oxidative phosphorylation. And so we know that electrons are uh, produced in the electron transport chain, where they are then passed to um, ATP synthase to produce ATP. Um, and so this picture basically uh, is the chemiosmosis model of the electron transport, the electron flow. Um, and for the sake of time, I'm not going to get into detail about like how all of these are happening. If you want that detailed description, go to chapter 19. Just email me, maybe able to. Yeah, let's see. So getting into ATP synthase, we want to talk about this, its structure and function, to understand how the alterations in cancer are happening through the impact of this enzyme. So the ATP synthase complex is said to be a molecular motor that converts energy associated with the proton motive force. That is said to be associated with the proton motive force into a rotational force that causes a conformational change that results in ATP synthesis. So this enzyme consists of two main parts. It consists of the F naught region and the F1 region. So this is the F naught region. This is the F1 region. So the F1 region is the catalytic sector of the active component of ATP synthase. Uh, so that is this region here. It has three nucleotide binding sites located on the matrix side. So those binding sites are the beta sites, and there's three of those. So it's a hexamer, but only three of those hexamers are what actually gets used for catalysis. So it is important to note that proton translocation through C units causes a rotational. It causes a rotation of the F naught subunit and the central shaft, which causes the conformational change within the three pairs of subunits, and the conformational change in one of the three pairs promotes condensation of ADP and PI to ATP. So basically, that's what's happening as we're rotating. Um, each of these beta subunits will go through a different conformational change. As those conformational changes happen, there's a different reaction with ADP that happens, with ATP that happens for the release of ATP, for the release of ATP. So, um, like I said, the beta subunits, which are one, two, three, undergo one of three conformational changes. So that is, it can undergo tense state, loose state, or open state. And like I said, all those involve different reactions of ATP. And it's only in the open state can ATP be released from the beta subunit open. and only in the open state can the ADP and inorganic phosphate bind. Okay, so that's kind of important to know there. Um, and so leads us to so our next slide is the F1 region, and so this is the membrane bound region, and it also includes the alpha subunit and multiple C subunits. And there's about 15 of those in human cells. And so it's the arrangement of these subunits that creates a channel through which protons can pass back and forth, as we see here. Um, when proton binds to a C subunit, it induces a conformational change that causes the C ring to rotate. That rotation depends on the neutralization of the negative charge of each asparagine and glutamine residue. Okay, so that's what we see here. It is the actual rotation. As the actual rotation occurs, the actual rotation occurs as the bound protein moves through the channel, and so it effectively pulls the C subunit along with it. Um, as the rotation takes place, the bound protein comes into contact with the subunit, where it is then released into the matrix. Um, the This proton will protonate this histidine residue in the subunit and transfer that proton to a nearby glutamine residue here. Okay. Um, and so it's important to note here that cancer cells, and so what we just basically described is how non-oncogenic cells or phagocytes operate, but cancer cells exhibit an altered metabolic pathway characterized by a preference of aerobic glycolysis or the Wahlberg effect. And so here is essentially where even in the presence of oxygen or the absence of oxygen, cancer cells can take up large amounts of glucose and convert it to lactate. So um, there have been studies that have shown that the increased production of lactate in oncogenes can lead to an acidic environment that thus promotes tumor invasion and immune invasion as well. So it is important to recall that the IF1 protein inhibitor can be associated with ATP synthase. We learned that in chapter 19 that IF1 binds to the catalytic sector of the F1 unit and can fully inhibit ATP synthase's activity at a low pH and low mitochondrial um, low matrix potential. And so, with that in mind, the literature displays the the literature displays investigations that were posed to understand the mechanism of the alterations in ATP synthase, its function, and its resultant contributions to metabolic reprogramming observed in oncocytes. And so ATP, so that brings us to our next, topic, which is ATP synthase and cancer, or the main topic, ATP synthase and cancer. So, ATP synthase and cancer cell proliferation. And so we know that can take place through the Warburg effect, which is what we just spoke about. We also know that inhibition of ATP synthesis, this is basically the blocking of ATP synthesis either directly with oligomyosin, which inhibits ATP synthase, or indirectly with retinol or antimycin, um, antimycin A, that is. This reduces proliferation in several cancer cells, including those derived from leukemia, blastoma, and certain cervical, lung, and osteosarcoma cells. Okay, and so you know, It's also important to note that um, cancer cell proliferation can be rescued by a uh, FCCP, which is basically a group of compound that functions as a proton ionophore and a mitochondrial uncoupler, which is based, and also the group basically described this as well in their paper. Um, in osteosarcoma cells, 143B cells specifically, oligomycin-induced inhibition of ATP synthesis, it blocked proliferation, um, which the effect can be reversed by the FCCP, which suggests that the uncoupling of electron transport chain from ATP synthesis might still support proliferation in certain contexts. But full oxos activity may be essential in tumors lacking efficient uncoupling mechanisms. So our next point is the mitochondrial respiration and biosynthesis of aspartate. So aspartate production, uh, it plays a crucial role in the ETC cycle. Uh, Proliferation by enabling the biosynthesis of aspartate. So uh, I'm sorry, I said that wrong. The the ETC cycle plays a role in cell proliferation by enabling biosynthesis of aspartate, which is, as we know, is a very important amino acid for cell growth. Um, and the group was able to come to this conclusion by using CRISPR-based negative selection screen in T cells. Um, They also found that supplementing the cultural media with aspartate or expressing an aspartate transporter um, could rescue proliferation in cells with impaired mitochondrial respiration, which highlights the importance of mitochondrial respiration in maintaining um, biosynthetic pathways for tumor cell growth. So for the role of ATP hydrolysis in tumor growth, ATP hydrolysis happens under hypoxic conditions. Um, in osteocarcinoma sarcoma cells, it is IF1 silencing, which basically prevents ATP hydrolysis. It is that silencing that causes ATP hydrolysis um, by the F1 component of ATP synthase under anoxic conditions. So the presence of the ANT2 transporter um, is said to facilitate the uptake of ATP, of the production of ATP through glycolysis into the mitochondria of hydrolysis by ATP synthase. So this process could potentially proliferate, I guess, potentially support proliferation under hypoxic conditions, but in certain liver cancers, um, the group found that there was an expression of lower levels of the ANT2 transporter, um, and the also found that the hypoxia results in G1 cell cycles was arrested, suggesting that the role of ATP hydrolysis in proliferation may vary across cell types. So that basically just means that like it depends on like I guess the uh, location of the cell or cancer and its environment. Like this paper kind of basically goes and you see how some cells have different like selectivity for different what word am I looking for? Like for different modulations of ATP synthase and get to that in the next slide. And so for the role of ATP synthase in cell migration, ATP synthase plays a role in cell migration. Um, and the group was able to make this comparison of primary tumors and lung metastases in 4T1 breast cells. Um, It was also observed that oxidative phosphorylation increased in circulating cancer cells compared to tumor cells. So this basically means that cancer cells with a higher mitochondrial activity such as those with increased oxidative phosphorylation, uh, showed more with increased oxidative phosphorylation and increased mitochondrial function, or um, or those with or those upregulated ATP synthase. They tend to exhibit more aggressive behaviors, specifically the ability to move or migrate away from their original location. So in the paper, an example given of this is the PGC1 alpha and ATP synthase. This is basically a transcriptional co-activator. Um, the PGC1 alpha, that is, and it promotes mitochondrial biogenesis. Uh, and that was found to enhance the oxidative phosphorylation in invasive 4T1 cancer cells. And so this effect was accompanied by the upregulation of ATP synthase alpha subunit transcript. So again, that's just the increased ATP synthase activity supports migration in metastasis. So um, um, and so I want to also talk, the paper also mentioned PGC1 alpha knockdown, and that's basically reducing PGC1 expression the in these cells to a decrease in respiratory chain complex gene expression and a reduction in cell migration. And the group was able to further link ATP synthase and mitochondrial activity to metastatic behavior through this specific um, investigation of the PGC1 alpha knockdown. And so it's also important to note that the authors in this group also proposed uh, that ATP synthase is a crucial player in formation and regulation of mitochondrial uh, uh, permeability transition or the PTP, which is basically a key component of apoptosis. Um, and so through structural interactions in ATP hydrolysis, ATP synthase could contribute to PTP opening, which influencing cell death pathways. And so targeting this pathway um, is an insight into enhanced therapeutic strategies. Okay, so here, so before we get into what this figure is showing, I kind of just want to talk about I just wanted to talk, okay, we actually are going to talk about this, but I'm sorry. And so basically what we're seeing here is basically ATP synthesis, synthase, localization and expression in in tumor cells. So this figure just basically shows the expression patterns um, in tumor cells of various tissues. And so through immunohistochemical staining um, of the ATP synthase subunits, it was revealed that various cancer tissues, um, it was revealed that in various cancer tissues that the activity of this enzyme specifically is highly concentrated in the mitochondria with enhanced expression. Um, and this is consistent with the enzyme's role in oxidative phosphorylation and ATP production. Um, it is also emphasized in the picture is also like emphasized in the picture the heterogeneous expression of ATP synthase across different cancer types. So um, it's highlighting the variability and its contribution to tumor metabolism. And an important thing to note is that in breast cancer, the reduction of the, re- the reduced expression of ATP synthase in metastatic lesions compared to primary tumors suggests a shift in metabolic needs during the metastatic process. And, and so all of these are just different subunit regions that the paper kind of goes into small details about certain cancers that are happening at these specific regions. Um, Especially this A6L. We will talk about that with soon, and the OSCP as well. So, which it's important to know that IF1 is a physiological inhibitor of ATP synthase. Um, And we know that the N-terminal region of IF1 binds to the catalytic subunit of ATP synthase, while the C-terminal forms dimers and tetramers. So, like I said, the N-terminal is going to bind to the subunits of ATP synthase, which we see here, right? Um, and the C-terminal is what forms dimers and tetramers, and that's what's depicted there. And so the binding of IF1 to ATP synthase is regulated by post-translational modifications. What are those post-translational modifications? Well, phosphorylation, um, which is, well, one of them is phosphorylation, which is influenced by the cell's metabolic state. It's also important to know that elevated IF1 expression is linked to poor prognosis in cancers, um, like small cell lung cancers, but other cancers like breast cancer and colon cancers have high IF1 levels and may predict better outcomes. So the group was able to use experimental models um, to to view IF1 overexpression. They found that IF1 overexpression promotes metabolic programming and enhances aerobic glycolysis, which contributes to cancer cell proliferation. And so specifically the paper itself um, um, was able to find that IF1 has been implicated in protecting cells from apoptosis by maintaining mitochondrial morphology. Um, so if we were to ask a question on what is an enzyme, which enzyme is contributing uh, to protection against apoptosis, um, in the literature, we were given an enzyme of OPA1 and DRP1. But it's also important to note that the role of IF1 in apoptosis resistance and cancer progression is complex, and we don't know everything about it. Okay. Um, and so the next slide is basically the expression of ATP synthase in cancer. And this has been shown to play a significant role in tumor development and progression. Um, Cancers often exhibit altered mitochondrial function. That uh, changes the way ATP is produced and um, the metabolism of energy. So ATP synthase, we know that that's a key enzyme in oxidative phosphorylation. It's crucial for cellular balance, and its levels can be modulated in response to metabolic demands of the cancer cell. So, uh, in some cancer cells, ATP synthase is overexpressed. Um, supporting the increased energetic requirements of rapid cell proliferation. So as this um, enzyme is overexpressed, it more ATP molecules are being produced, thus driving proliferation in these cells or I guess enabling these cells to grow rapidly. And so it's important to note that this overexpression is linked directly to metabolic programming. What is that programming? So that it includes the shift towards aerobic glycolysis. And additionally, the expression of ATP synthase can influence mitochondrial dynamics, um, like the membrane potential, how many ROS species we're um, producing. And both of those play a role in um, the survival of cancer cells, its proliferation, and also its resistance to apoptosis. So the regulation of ATP synthase expression and its activity emerges as one of the most important factors in cancer biology and therapeutic strategies. And so the figure that we're seeing here um, is basically just shows the impacts of expression on metabolic programming in cancer cells. So, like I said, the overexpression of ATP synthase can influence energy balance in cancer cells. Um, and Also, the figure just shows that high ATP synthase levels correlate with increased ATP production through oxphos. And also seen here, um, high levels of ATP synthase can also, uh, sorry, decreased levels of ATP synthase can lead to decreased oxidative phosphorylation and uh, like a complementary increase in glycolytic activity. So uh, this would be the downregulation of ATP synthase, and this would be the upregulation of ATP synthase. So when ATP synthase is downregulated, say in OSCP, this is mostly found in colon cancers. On the opposite end, the upregulation of OSCP is mostly associated with stomach and breast cancers. And so what the group was trying to do is just basically uh, kind of compare how the downregulation of certain subunits within this enzyme has a different effect on cancer cells, say when the enzyme is upregulated. That's basically what that whole figure is describing here. Okay, so it's also important to note that mutations, that ATP synthase can undergo mutations and modulation. So we've kind of talked on the metabolic rewiring, and so we're going to touch on that just a little bit. We're going to talk about some resistance, therapy resistance, and modulations of ATP synthase and a specific epigenetic modification. So for rewiring, um, we know that cells can undergo metabolic reprogramming, which is influenced by epigenetic changes. Um, ATP synthase can also be resistant to therapies. So book the group gave an example of ATP5B and OSCP were found to contribute to chemo resistance in cancers like chronic myeloid leukemia, colon adenocarcinoma, and HER2 breast cancer. Another resistance example that the literature gave was the hypermetallation of ATP5B, which is basically a promoter that leads to the downregulation of ATP synthase beta subunit. Um, and this was found to be implicated in disease progression and drug resistance. So with the when talking about the subunits and resistant mechanisms, ATP5B downregulation was linked to adrenomycin resistance in leukemia. Uh, And in colon cancer, the loss of ATP synthase subunit D correlates with resistance to 5-FU. Um, it also correlates with the increase of reactive oxygen species and a poor survival rate of those cancers of those cancer cells. Um, and so S the SC the OSCP subunit downregulation in colon cancer. The group found that this was linked to the resistance and evasion of apoptosis via inhibition of mitochondrial um, of the PTP, and um, and so it's and oh, my bad, Lord have mercy. And so for the modulations of cancer treatments, the group was able to find an increased expression of ATP synthase subunits of the beta and the gamma subunits in first breast cancer. Um, Found that those were resistant to trastuzumab therapy, which is the therapy for breast cancer. And so I would like to end on talking about the epigenetic modifications. And so the group were able to find that epigenetic regulation of ATP synthase subunits, whether down or upregulated, is a key mechanism of understanding their therapy resistance in cancer. Um, and so through through the modifications, well, through the studies, the group was able to provide us with um, key insights on ATP synthesis function in cancer. And I think this is very, very important for anyone who's interested in oncology or interested in um, immunology or pathology. There's not a lot of research on this specific on these specific processes, and so I think this was very interesting to read upon and learn about, and also be able to present to you guys. And so this is just a conclusion. So we basically discussed on the critical role that ATP synthase has in cancer metabolism. We discussed the epigenetic regulation of ATP synthase. We discussed how therapy resistant mechanisms take place and potential therapeutic implications. And so I have a few questions. The first question is, besides the modulatory effect of IF1 on ATP synthase, describe another mechanism that promotes the rewiring of metabolism in human carcinomas. The next question is, what is the significance of the protein OPA1? And is it associated with apoptosis? And that is all.